one you can see is a little bit more of an acute angle. Here's one that's a little bit more of an obtuse angle. That may represent the natural flexibility of the two arms with respect to each other, but we also have to worry that there could be some distortion of that angle upon drawing the molecules down onto a surface to do the electron microscopic experiment. So currently we haven't come to a clear resolution of what the breadth of this distribution means, so we pay more attention to the angle between the two arms. So being encouraged then that this idea of extending the arms of an RNA might be able to give us the angle between the arms in the folded structure and convert an intrinsically low resolution technique to give us a higher resolution view of what's happening in the middle of the molecule, we now go back to the ribozyme. Here's a sort of a road map of the tetrahymena intron with the different helical elements shown. And from previous work done in our laboratory, we figured that extension of this arm called P6b, an extension of this paired region called P8, would not interfere with the folding of the molecule. And we, in fact, have an easy way of testing this because we took the molecules with these helical regions extended, used them as catalysts, did the sort of detailed monitoring the rate of ribozyme catalyzed cleavage, and we directly showed that extension of these two arms did not perturb the function of the molecule, and therefore it seems to be neutral with respect to the folded structure. So we can then use these extensions as reporter arms. To figure out what the angle between these two helices would be within the folded molecule. What would we expect? Well, from the French model, that of Michel and Westhoff, shown here, if we extend the P8 region, which is this yellow portion, as a helix, and 7D base pairs is in fact much longer than this, but we ran out of room on the slide, so I'm just showing you the first turn and a half of the helix, which is about the first 15 base pairs, it would go out in this direction. If we extended the P6 region, shown in blue, it would go straight down. And again, the expectation, as in the case of tRNA, would be a 90-degree angle. We did the experiment. Here are ribozymes seen in the electron microscope with all at the same magnification. It's a little lower magnification than I showed you before. Here are ribozymes in their natural state with no helical extensions. Here are ones where we've extended a single helical arm. And now in C and D are ones where we've extended both the paired region 6 and the paired region 8. And you can see that they map out a distance, an angle, which when we measure some hundreds of these molecules, we find out is right at the 90-degree range. We've done this also for pairwise extensions, not only of P8 and the P6 region, but also for extended other helical regions, and we can then get a map of how these helices, what angle they form relative to each other in the folded RNA molecule. Now, the title of this slide is Chirality Strikes Again. Chirality is a Greek word for handedness, and it just has to do with how your left hand relates to your right hand. They have a mirror image symmetry. It's very common in biological molecules for the molecule to be, and it's either the molecule or its mirror image to be the active form. And I'm just pointing out here that from these electron microscopic experiments, we cannot distinguish whether the structure that we've determined is the one on the left or its mirror image shown on the right. From other experiments, it looks like the ribozyme is probably in the form shown on the left. This shows that some scientists like to be artists in their spare time, so our collaborator Jack Griffith decided to digitize some of the images of these ribozymes with their helices extended and arrange them on a red background to make an artistic view of RNA structure. The editors of the journal where we submitted the paper wanted to know whether they would destroy the scientific validity of this image by turning it upside down, and I assured them that there was no scientific meaning to this particular arrangement of the molecules chasing each other around in a circle, and so that they could put it on the cover of the journal in any orientation that they wanted. Finally, it's worth saying that this helix extension model, although sort of a fun thing to explain to you today, is a pretty low resolution technique, and we are trying to achieve a level of resolution where we can see every atom in the molecule in its correct location at very high resolution. This has been done for the transfer RNA molecule. That beautiful picture I keep showing you, that's sort of the purplish color, was determined by X-ray crystallography. That has been the world's record for the largest RNA molecule that could be, whose structure could be completely determined by growing crystals. And you know, in a crystal, the molecules are all lined up in the same orientation, one right after another, so that when you shine an X-ray beam through that ordered array of molecules, then from the diffracted spots of the X-ray, you can reconstitute a picture of what the individual structure of each molecule must have looked like. So we've been working very hard to grow crystals of ribozymes or large portions of the ribozyme, and you can see that from this picture that we've had some success, and in fact in the last month we've had a breakthrough in the solution of the crystal structure of a large domain of the ribozyme, about twice the size of transfer RNA, in collaboration with Jennifer Doudna's laboratory at Yale University, where Jennifer was a former associate in the laboratory, and we've now that she's gotten her independent faculty position, we continue to work on this project collaboratively. So hopefully within the next year you will see a domain of the ribozyme in a scientific magazine of some sort, portrayed at the level not just of the angle between helices, but at the level of where the individual atoms are actually located. And so thank you for your attention, and I'll be glad to take questions. Yes?
Are there any inhibitors that fight for the active site with the ribosome?
So the question is, are there any inhibitors that compete for binding to the active site of the ribosome?
Either change in conformation or, as I said, fight for the active site?
So we've investigated the possibility of inhibitors, in part because there are some pathogenic organisms that have these ribozymes, and it might be interesting to ask the question, could you inhibit this pathogen by interfering with its self-splicing RNA? So far we've found two separate kinds of inhibitors that I think make a lot of sense with respect to the mechanism that I showed you. One is a guanosine molecule that is similar enough to normal guanosine that it can bind into the guanosine binding site, but it doesn't have a 3' hydroxyl group, this OH group, so that it can do the attack. So that sort of an inhibitor does shut down the reaction, but it has to be used at very high concentrations, so I'm not sure that it's in order to fill up the binding site. So I'm not sure that from a medical point of view it would be very useful. This is a bit similar to one of the HIV drugs that is being used, which is the AZT, the azithromycin or thymidine, which is something that is one of the enzymes of the virus is fooled into using instead of a normal nucleotide, but then it can't do the correct reaction. And so that's the same sort of idea, that any catalyst that utilizes nucleotides